And we're really happy to have you all join us for today's session with Elizabeth and going over a primer on GWAS and a little bit more about her recent work with Tractor. So Elizabeth, turning it over to you.
So yeah, given the Stanley Center has such a diverse sort of set of research expertise, and there's a lot of new folks who've been joining recently, I was sort of tapped to give this intro talk to kind of serve as a leveler to make sure that we're all kind of on the same page, you know, in terms of understanding GWAS and interpreting talks, you know, moving forward throughout the seminar series. So that's kind of the intention of this talk. So I'll be starting off with a kind of intro to some of the more basic concepts related to GWAS, the goals, and then unpacking some of the actual math and nuts and bolts of GWAS, as well as how you generally present and interpret results. And I expect that this kind of intro primer piece will take about half the session. And then I'll kind of pivot to a deeper dive into a specific flavor of GWAS that I've been developing recently, which is kind of has the goal of enabling well-calibrated association studies on admixed populations. So that'll be sort of another half hour. And then the plan is to break into breakout sessions to discuss, you know, questions related to standard GWAS, which will be led by Hailey Yang, and questions related to Tractor, which I will host. So that's the lay of the land for the next 90 minutes. So thanks for calling in. Okay, so let's get into the GWAS primer, starting with a very big picture view. So let's definitely start off with a definition of GWAS. So a genome-wide association study is an approach used in genetics research where we associate specific genetic variants with particular diseases. So this method involves scanning the genomes of lots of different people, looking for genetic markers that can be used to predict the presence of a disease. And then once we've identified these markers, we can hopefully use them to develop better prevention and treatment strategies. So I really like this quote that I put on the screen from Francis Collins that I stole from the landing page of the NHGRI. And it sort of describes the power of GWAS. He points out a few things that I think are pretty good to keep in mind in a nice and concise way. So the first is that you need large sample sizes to have good power. He also notes that it's vital to ensure kind of a similar composition in your, for example, cases versus control cohorts. You, of course, scan across the entire genome, testing each variable position for an association with the phenotype. And this is how you're able to kind of hone in on a particular spot in the genome associated with the phenotype of interest. He also points out a few other considerations, which are that this is a hypothesis generating sort of approach. So you don't know ahead of time which areas are, you know, going to be found to be associated. And you're also not limited to just looking at genes that have been previously associated with your phenotype of interest, as we had been in previous candidate gene studies. He also notes that you need to be really careful with your statistics to avoid false positives. This is something that I'll come back to repeatedly throughout the talk, in particular with respect to considering ancestral diversity in your GWAS. So GWAS have really revolutionized the identification of genomic regions associated with complex diseases. They've identified thousands of robust associations for hundreds of complex traits. And just to note, these numbers are really orders of magnitude bigger than the replicable linkage and candidate gene association findings to date. So really have provided a major boost to the field. So here I'm just showing the kind of GWAS epidemic, if you will, over the years. So starting in the early days through today, you can just really see this massive growth in the number of publications involving GWAS. And viewed a different way, if we look starting at these early days of GWAS and partition it now by how many genes have been associated reliably with various diseases, we can also see this increasing dramatically. So here we have from 2006, all the known disease genes, which are indicated as these lollipops off of the chromosomes, color-coded by the kind of trait that this association was for. So if we go forward in time, you can see by 2008, we're already starting to find many, many more disease genes. They're also starting to pile up in some of the same locations, and you'll notice that some of the same spot is starting to get associated with multiple different colored lollipops. So there's some pleiotropy already being seen. By 2010, things are starting to get even more crowded. By 2012, the plot has to start getting expanded lengthwise because there's so many associations. And here's the last figure like this I could find from 2017, where you can see there's over 13,000 SNP associations, and it's becoming a very sort of dense landscape of known genes associated with complex traits. So we're really have a whole ton of GWAS results swarming in, and these are continuing to just sort of increase in numbers. So why do we keep finding more and more associated variants, despite the fact that some of these studies are done on even the same trait or on very similar traits? This gets into the kind of genetic architecture underlying many diseases. So rather than being Mendelian, where a genotype at a single locus generally can be thought of as causing a disease, most human diseases have been found to be polygenic. So this means that many, many loci spread across the genome all contribute in part to producing the phenotype. So rather than just one locus of really big effect producing the phenotype, there are many, many loci of very small effect that together contribute to influencing a person's disease status. And that's what allows there to be this kind of smooth bell curve of normal variation for many phenotypes, as you can see from this review figure that I put up here. So since these variants are each individually contributing only a very small effect to the phenotype, you need to have a really big sample size to be able to detect them. So as sample sizes have grown and grown over the years, we're continuing to find more and more hits even for the same phenotype. So we've seen how many findings we have. Have we found them all yet? Are we near GWAS saturation? Survey says no. If we actually look at the paths of how many GWAS hits we see for different data freezes of the PGC collections, these are these dots along this figure here, we can see that we're still in kind of the linear increase in association stage. So based on the sort of hypothetical expectations for finding significant GWAS hits, initially we would expect this dead zone whose length depends on how many cases have been accrued and the largest effect size for that particular phenotype. Then we'll expect this sort of inflection point where we have sort of a big enough sample size to start to find significant hits, followed by this linear increase. Once we've started to find most of the hits, we'll expect this kind of diminishing returns plateau to happen. But as you can see for these psychiatric phenotypes, we really seem to still be on this linear increase. So that means there are still many more variants to be discovered that will be found as we continue to boost our sample sizes. Another thing I'd like to note at the beginning of this talk is that different genetic strategies are better at detecting different types of genetic variation. So we often conceptualize disease associations in two dimensions, the allele frequency and the effect size. So highly penetrant alleles for Mendelian disorders with extremely large effects that are pretty rare are kind of in this upper left quadrant, where you'll notice that most GWAS findings are associations in this bottom right. So common SNPs that together have pretty small effect sizes. This is kind of the area of the spectrum that GWAS are sort of best tuned to find. Okay, so that was our big overview of GWAS. Let's get into the methodology a little bit. So what is a GWAS exactly doing? Well, once we have a well-defined phenotype and genotypes have been collected using sound techniques, we can run an association to connect the phenotype to this particular spot in the genome. And the de facto analysis for GWAS data is generally a series of single locus statistical tests. So we'll examine each SNP independently for an association to the phenotype. The specific test that we conduct depends on a variety of factors, but sort of first and foremost, they are going to be different for quantitative versus case control traits. So this figure shows examples of data from both categories, a case control setup at the top and a quantitative trait biomarker at the bottom, and they're both partitioned by the genotypes of the component individuals. So you can see in the case control example, each person is just going to be color coded by their case status. Are they a case? Yes, no, basically. And if the SNP were to be associated, you would see sort of different frequencies of cases versus controls in the different genotype classes, AA, AG, and GG. So the GWAS in this case will be basically evaluating differences in the proportion of cases and controls between these genotype classes for this given SNP. And that's what allows us estimation of the association of that SNP with the risk of disease. So instead, in the quantitative traits case, the identification of the variance happens instead of classifying people ahead of time by their status, by looking at the level of the association of a continuous biomarker by comparison of their biomarker levels by genotype class. So rather than, looking just, like I said, assigning people ahead of time, we're actually going to look at the means of our, biomarker quantitative trait of interest and see if the means are different depending on the genotype that the person has. So quantitative traits, as I mentioned, are generally analyzed using a generalized linear model approach, commonly the analysis of variance, which is similar to linear regression, except for we have categorical variable predictors. So in this case, this is going to be the genotype classes. And I'll often be using just the shorthand of calling this linear regression throughout the talk. And just to be very clear, the null hypothesis for this test is that for a single SNP, there's no difference between the trait means in any genotype group. So we're testing that null hypothesis against the alternate hypothesis that the genotypes means are different depending on your genotype status. I also want to note that the assumptions for this is that, there are several assumptions for this that are important to consider. One is that the trait is normally distributed. The second is that the trait variance within each group is the same. And the third is that the groups are independent. So these are really important things to consider when you are going to be running your GWAS. And we'll talk about other considerations throughout the talk as well. So turning to the top half of this slide, so for dichotomous case control traits, as I mentioned, we're generally using logistic regression or sometimes contingency table methods to analyze them. So logistic regression is basically an extension of linear regression, which I'll go over in the next slide, where the outcome of a linear model is transformed using a function that predicts the probability of having a case status given your genotype class. This is often used for case control settings. It also has the nice features of allowing for adjustment for clinical covariates, and it also can provide an odds ratio as a measure of effect sizes. All right, so let's get into the model. So most researchers will generally assume an additive model when they're running their GWAS. The additive model assumes that there's a uniform linear increase in risk for each copy of the allele, as you can see in this figure. So 2 copies of the risk allele will double your risk as compared to being a heterozygote. The additive model is often the only one that's tested in GWAS studies. It's been found to have reasonable power to detect both additive and dominant effects, but it is important to note that it can be underpowered to detect some recessive effects. So the basic GWAS model is the one that I'm presenting here, where we test each SNP for an association with the phenotype using the additive model, where Y is a vector of our observed phenotype values for each person in the study. This beta 0 is our intercept term. The X is going to be the number of copies of the minor allele for each person at that particular SNP. And beta 1 will be the effect estimate for the minor allele at that SNP. And then we can also put in this epsilon, which can capture our error. So this is really just the core nuts and bolts of GWAS. You can expand this model in various ways, but this is really the kind of main infrastructure that's good to understand. So what do you get out of this test? You generally get 3 main pieces of information. You get an estimate of how strongly associated the SNP is to the phenotype. So this is your p-value. You get an estimate of how much of an effect the genotype has on the phenotype. So your beta, your effect size estimate. And you get an estimate of the variability in that effect. So can we just run our GWAS and then take the results at face value? Can we always assume that getting a really strong p-value means a really good result? No, of course. There are a variety of reasons aside from a true effect that can cause very strong associations with the phenotype, and these are really important to check and control for. So such situations can result in false positive associations due to either just simply chance, some kind of bias or confounding in your study, or other reasons. So for that, we do very stringent QC. We do careful correction for multiple testing. I'll go over both of these in a moment. And we examine our GWAS associations after the fact to ensure that we trust them. So in terms of things to be careful about going into your GWAS, the first key consideration, as I alluded to, is to have good and consistent phenotyping across cohorts and classes. So phenotype QC should be done beforehand to make sure that you have good repeatability for your measurements, good inter-rater agreement for phenotype consistency, and that you've removed any clear outliers that were just simply mistakes entering the data. Because we want to make sure we're finding variants that are truly associated with the phenotype and not, batch effects from different collection centers or something. Likewise, you should do very careful QC of your genetic data to make sure you're getting rid of variants that represent sequencing errors, as these can also cause false positives in your results. There are a number of pipelines in place and frameworks for recommendations for doing good genotype QC. These can vary a bit depending on your study design and type of genetic data. But members of the Stanley Center have actually curated some very good resources to inform your variant QC. So I'll point you to those.
Elizabeth, can I ask you a question about the standardized phenotype criteria? This, of course, is a really difficult question for psychiatric diseases where there are no objective ways to measure whether someone has one diagnosis versus another, and diagnoses change over the life course. What are ways that you as geneticists think about this thorny problem for psychiatric disease in particular?
Yeah, so I guess there's a few ways. One is we rely very heavily on our clinical colleagues. I myself, I'll be first and the first to admit I'm a geneticist and not a clinician. So I'm definitely going to be leaning heavily on the expertise of people who are trained to diagnose these challenging and related phenotypes. In terms of just data-wise, people often do check for example, like inter-rater reliability. So they'll have multiple different clinicians rate the same person and make sure that there's agreement in their phenotype assignment. And yeah, just looking at the distribution of phenotypes, you can see if one collection center is having a different distribution than another, this can provide some hints and if there's some systematic difference. But yeah, it's a really challenging issue, and definitely rely very heavily on our clinical colleagues to help inform the best phenotyping strategies.
Great, thank you.
For sure. So yes, QC is very important for every aspect of our data. We can also, getting back to the modeling, we can control our statistical tests or adjust them for factors that are known to influence our traits, such as age, sex, the study site, or known clinical covariates. So by adjusting for covariates, we can reduce spurious associations that are due to these potential sampling artifacts or any biases in study design. It is important to note that adjustment comes at the price of using up additional degrees of freedom, which can impact your statistical power. And just as a teaser for the second half of the talk, one of the more important covariates to consider in genetic analyses is a measure of population structure. So I'll just sort of introduce this concept now, but we'll go into a much deeper dive in the second piece with Tractor. So there are often known differences in phenotype prevalence due to ancestry. There's also known allele frequency variability across human subpopulations. So this means that in a sample with multiple ancestries, ancestry-specific SNPs can likely happen to be associated with the trait just due to this structure, also known as population stratification. So just said differently, because of naturally differing allele frequencies across ancestries, if you don't account for the ancestral makeup of your samples well, you can get spurious associations at variants that just partition ancestral groups rather than that are truly related to the phenotype. And I'll go into this, as I said, in much more detail later on, but I thought it'd be good to start sort of building intuition now, especially as we're going to talk about how to examine if we do have a problem with substructure in our GWAS results. So these potential false positive associations from population stratification will mean that there are going to be a ton of positions across the genome that will appear to be highly associated with the phenotype. So how do we know if we didn't correct for all of the population stratification in our sample with all the covariates that we put in? Luckily, we can examine our GWAS results after the fact to see if they look okay or show signs of harboring any population structure. And we typically do this using a QQ plot. So the QQ plot is a graphical representation of the deviation of the observed p-values from the null hypothesis. So basically the observed p-values for each SNP are sorted from largest to smallest, and you'll plot them against the expected values from a theoretical chi-square distribution. So if the observed values correspond to the expected values, all points should be near this line in the middle here between the x-axis and the y-axis, and this would be the null hypothesis. There's no SNPs that are looking significantly more associated with the phenotype than expected. If some observed p-values are clearly more significant than expected under a null hypothesis, they'll move towards the y-axis. So if there are not so many of these, so on the kind of right-hand panel, you'll see we're sort of following the null hypothesis, and then there's this handful of SNPs that are lifting off as being more significantly associated than expected, then it's plausible that these could be the true genome-wide significant hits. However, if there's an early separation of the expected from the observed, like we see in this middle panel, this means that many of these moderately significant p-values are much more significant than expected under the null hypothesis. And this is rarely due to, many thousands of true positives, although, for some phenotypes, we are getting the sample sizes where we need to start thinking critically about this. But in general, this is more often likely to be due to population stratification, as I described in the previous slide. So just again, to reiterate this, systematic differences in your allele frequencies between subpopulations will cause many more p-values to appear to be strongly significantly associated than would be expected from chance alone. And that's what this inflation is reflecting. So you'll also generally see a lambda score reported with your QQ plot. This is sometimes called the genomic control lambda, or lambda GC. So genomic control estimates the overdispersion in your test statistics on that increase in the median test score by the inflation of the association scores beyond what one would expect under the null hypothesis. So it's basically putting a value on sort of the shape of your QQ plot. So it's going to assume that overdispersion is constant across the genome, and it basically is the ratio of the median observed test statistic to the expected test statistic. So yeah, just a useful sort of summary measure of the amount of dispersion of your test statistics. And in terms of interpretation, a lambda of 1 means your test was adequately controlled for population structure. If you have one that's substantially above 1, that would indicate that you have a problem with inflation, and you should probably go back and try to correct for population structure in your data set. Another important consideration to processing your GWAS results is correcting for multiple comparisons. So as you know, statistical tests are generally called significant if their p-value falls below 0.05 as the alpha value. So this would mean that about 5% of the time, the null hypothesis is rejected when it is in fact true, and we would find a false positive. The issue, though, is that this probability is relative to a single statistical test. And in the case of GWAS, we're testing hundreds of thousands or millions of tests of SNPs. So we're conducting, you know, millions of potential tests, each one with its own false positive probability. So the cumulative likelihood of finding one or more false positives over the entire GWAS analysis is therefore much higher. And in fact, if we just use the standard 0.05 with a million tests, we'd get 50,000 false positives, which is obviously unacceptable. So to adjust for this, we correct for multiple comparisons. And the simplest way to do this is just to divide the alpha of interest by the number of tests conducted in a Bonferroni correction. So we generally assume that there are about 1 million independent LD blocks in the genome. So this is the effective number of tests that we're going to consider if we're doing a genome-wide analysis. So about 1 million independent tests will happen in your genome-wide scan. So we'll divide our alpha by 1 million, and that's where we get this 5 times 10 to the negative 8 value that has become kind of the standard threshold used to designate genome-wide significance. And you'll see that number used most commonly to designate significance in the literature. Okay, so let's talk about presenting the results of GWAS. So the classic way to present your GWAS results is, of course, the Manhattan plot. So named because it sort of looks like the Manhattan skyline. The Manhattan plot illustrates two properties of GWAS results. The first is the physical location of SNPs with extreme p-values. And the second is the degree to which a SNP association is corroborated by other nearby SNPs in linkage disequilibrium. So this, I guess maybe just to sort of back up, what we're actually going to see here is the position across each chromosome where these SNPs are found, with the p-value, that's on the x-axis, with the negative log 10 generally, p-value shown on the y-axis. And you'll usually see this standard 5 times 10 to the negative 8 significance threshold listed. And often top hits are annotated with either the variant name or gene name. As I mentioned, you can sort of look at a top hit and see if it's corroborated by nearby SNPs, which would appear as a kind of peak in the Manhattan plot. So these are all SNPs that are very close by to each other on the chromosome that are all coming up as extremely significantly associated with the phenotype. And this lends credence to the signal being real. Whereas if you had kind of a lone hit that doesn't have any LD friends also finding an association, that would be a little bit more suspicious. And I'll be showing as examples of interpreting plots, real results from our recent PGC PTSD paper.
Elizabeth, can I ask a few more questions about that Manhattan plot?
For sure.
I noticed that the first couple chromosomes have very fat bases. There's many, many dots, and then later ones are very thin. Is that something particular about PTSD, that just by chance the first few have more associations? So that's actually a factor of just the chromosome length. So the first, chromosomes are kind of roughly ordered by their length. So the first few chromosomes just have more SNPs on them. So we have a longer, thicker portion of the x-axis devoted to those early chromosomes just because they are longer. They're bigger. Mm-hmm. And then in this plot, which I haven't seen in others, or maybe I wasn't looking close enough, there are these two deep valleys in chromosome 1 and chromosome 9. What are they doing there? Yeah, so sometimes you'll see these centromere regions. These are like basically areas where there were no SNPs. Speaking broadly, these were areas in the chromosome where there weren't really any SNPs to plot. And generally they're because of centromeres. Mm-hmm. Thank you. Yeah. Yeah, thanks for the questions. Cool. Okay, so if one were to go online and download a set of GWAS summary statistics, the results would generally be presented in a table that has some standard information, and I'm showing kind of an example of such a set of summary statistics that is sort of with this classic presentation.
So usually the first few fields are information about the variant. So we have this SNP ID name. Usually there is some information about the chromosome, maybe a chromosome position. Sometimes it'll say the closest gene. And usually some kind of information about the major and minor alleles and their relative frequencies. You can see that this sort of initial chunk is devoted to that variant information. Next, you'll generally see information about the association results. So this will include the effect size, maybe the odds ratio, and some kind of standard error estimate. And then, of course, the p-value for the strength of association of that variant to the phenotype. And then other information is often stuck afterwards. So this might be, you know, information sort of specifically about that particular study, maybe the sample size, other significant markers in the region, or in the case of a meta-analysis from a number of different studies, there'll often be an estimate of heterogeneity, which will tell you how consistent that result was across the different studies. But this is basically, yeah, what you would see if you download your GWAS summary statistics. Not too intimidating, hopefully.
Hi, Elizabeth. Can I ask
Yeah.
a question about that?
Of course.
Is there are there any standards by which the effect size, for example, or the odds ratio are reported with respect to like case versus control or minor versus major allele? Because I see, for example, in this table, allele 1 frequency, sometimes that's the major allele, sometimes it's the minor allele. And then how do I interpret the odds ratio?
Totally. Yeah. So I think that's the orientation of the alleles can differ. In this example, it was oriented to the effect allele. So I think in general, like, I mean, to be honest, this might vary across studies. I'm not 100% certain this is sort of a hard and fast rule, but I think generally they try to be oriented that way. Sometimes you will see them oriented minor, major. But yeah, definitely an important thing to check for interpreting results, because I totally agree this has bearing on how you downstream analyze these data, especially if you're trying to meta-analyze across various cohorts or something. So definitely a good thing to double check is that the orientation is the way that you think. Yeah, good point.
So I guess just to complete that
Yeah.
thought, like I guess in this case, you would ignore, you wouldn't worry so much about which was the more common or less common allele. You would just look at the odds ratio and say, if it's above 1, the allele shown here is more frequent in cases.
Yes, exactly.
Yeah.
Yep. Okay, so in terms of thinking about these types of results, I wanted to bring up another point. So can we just look at the top hits of our GWAS study and assume that they are going to be the causal variant? They're the most strongly associated with the phenotype. Maybe they're directly affecting the phenotype. And the short answer is no. So because we have LD across the genome, this presence of this LD creates two possible outcomes from a GWAS study. So in the first outcome, the SNP influencing the actual phenotype or the biological system itself is indeed directly genotyped in that study and found to be statistically associated with the traits. This would be sort of this red disease risk SNP here. If we actually genotyped this exact SNP and found a strong association with our phenotype, then in that case, yes, that would be the causal SNP, and that would be called a direct association. The second possibility is that the influential SNP is not actually directly typed, but instead a tag SNP that's in high LD with it is typed instead. So therefore, this other genotype SNP, this tag SNP, is going to be statistically associated to the phenotype just because it's tagging the true causal SNP. And this is referred to as an indirect association. So because there are these two possibilities, a significant SNP association from a GWAS can't be assumed to be the causal variant directly. and would require additional analyses, such as, for example, fine mapping to figure out the precise location of the causal SNP. And indeed, as you can see from this figure, most GWAS SNPs identified are not themselves coding. Some have been found in coding regions and in regulatory regions that could potentially affect gene expression, but many remain pretty mysterious in function. And this has been talked about previously in this session, and I think will probably be a major focus of this seminar series going forward. But this is one of the hardest challenges related to GWAS is turning GWAS findings to actually biological mechanisms. So that's out of the scope of this talk, but I want to note that that's a major area I think we'll be hearing about throughout this year. Okay, and we are now at the turning point. So that was hopefully a sort of overview to get everybody's feet wet with respect to GWAS. Now I'm going to turn to switch gears and turn to a deeper dive into a GWAS method I've been developing that allows you to do association studies in admixed populations. And I'll also discuss some of the implications of GWAS in diverse populations more generally, kind of echoing some of the themes from the MPG talk this morning. All right, so to start off, I'd like to reiterate a point that is now becoming kind of common knowledge in the GWAS community, and that is that the vast majority of association studies are conducted on European cohorts. So if we look, further down, even in this sort of small wedge of the pie representing, about 20% that are not European, there's only a handful of percent within that wedge that are actually populations of admixed origin. So this includes Hispanic, Latino individuals, African American individuals, groups who collectively make up more than a third of the US populace, but are only a few percent of GWAS studies conducted currently. And I think it's important to note that there's actually many more admixed individuals out there who have already been collected, genotyped, phenotyped, but are not making it into this figure. And often they're being intentionally excluded because of being admixed. There's also some important, significant efforts that are ongoing trying to collect more representative samples, including many admixed populations. This includes, all of us and some of the other efforts that, the Broad and Stanley Center specifically are heavily involved with. But historically, we really haven't even tried to consider our admixed samples. And this is, there's a sort of historical reasons for collection, but there's also a lack of methods to appropriately analyze diverse and admixed populations. So there's really sort of a pressing need to allow for easy incorporation of admixed people into association studies so we don't leave all this data on the table. And furthermore, there's other problematic aspects of leaving entire ethnic groups understudied. So hearkening back to Alicia's talk this morning, and also if you think back to the burst of GWAS publication results I mentioned in the first half of this talk, the sort of sad news is that if we color this rapid increase by the ancestry component, the ancestry designation of the participants, you can see that it has been really heavily driven by European individuals. So again, this is problematic for a few reasons. One simply being that this is not reflecting, equitably the global population, but we're really leaving a lot of data on the table and potential for discoveries on the table. There are some pretty clear benefits to including more diverse populations in GWAS. The first is that different populations will have different sets of variable positions across the genome, as you can see in this sort of classic figure from the 1,000 Genomes Consortium. So because of this, different variable spots across the genome in different groups, we are afforded the opportunity to associate our phenotype to different spots in the genome and different ancestries. So we have the potential to discover new loci that will be, helpful to understand for health for everyone, but that might be undiscoverable if we had restricted ourselves to just one ancestry. I'll also point out, particularly with respect to African descent populations, that if we look at these, this is sort of the number of variants per genome for different groups in the 1,000 Genomes Project. These yellow African populations have almost 1 million more variants than non-African populations. So there's even more spots across the genome that can be informative for disease in African descent populations. And there are some efforts led by the Stanley Center that I sort of vaguely alluded to before, including NeuroGap, who are hoping to start to probe this important source of genetic information.
Elizabeth.
Yeah.
On that point, are there enough more variants in those populations that you actually have to revise your correction for multiple comparisons? Because you said that's based on an assumption of 1 million LD blocks.
That's a great question, and that's something that actually we've done a little bit of digging into. So as part of our tractor manuscript, we did actually do an explicit test to figure out what the right sort of p-value threshold would be for, in our case, African American individuals. But yeah, that's definitely it is true that there are different numbers of sort of independent linkage blocks in different populations, and specifically there's going to be less linkage within Africa. So yeah, actually Masa Kanai, who helped out with this sort of portion of this talk, has a publication on this, and indeed, it seems there should be probably a slightly lower threshold in African populations compared to non-African ones. And actually for our admixed populations, we found that like 1 × 10⁻⁸ was probably good, something around there. Not substantially lower, but a different number.
Cool, thanks.
Yeah. Yeah, thanks. I didn't put that slide in here, but yeah, good question. Okay, cool. So one other quick point about the benefits of diverse populations before I get into the tractor method itself. And again, this was alluded to this morning. By looking at different ancestry groups, we can often do a better job at honing in to where the causal variant may lie. And this is related to a number of fine mapping efforts led by Hai Liang, who's kind enough to provide me with this slide. The overall idea is that the kind of patterns of linkage disequilibrium will be different in different populations. So if we assume that there's a shared causal variant that's going to have an effect on people of all ancestries, we can look to see where they kind of overlap to hone in much more finely on where the causal variant is likely to be. So by using information from multiple different ethnic groups, we can do a better job of understanding where the sort of causal mutation is for, again, all populations. Okay, so I want to circle back to admixed populations in general. So why are admixed people removed from GWAS studies? So they're generally removed due to the challenges of accurately accounting for their complex ancestry, such that population substructure and stratification can seep into your analyses and bias your results. And in the context of GWAS, as we discussed in the first half of this talk, this means that there's a potential for false positive hits due to ancestry rather than a real relationship to the phenotype. So I like this review figure from actually quite a while ago, but I think it's still very informative, that kind of shows this very clearly. So because different populations have different demographic histories, many sites across the genome are going to be at different frequencies in them. And you can think of this as being related to the different color-coded genotypes shown kind of in this key in the bottom of this figure. So if we're hoping to do a GWAS on a disease that has cases and controls, as we are in this example, and we just sort of naively grab individuals from population 1 and population 2, if we were to run an association at this particular SNP, we would highly likely end up with a positive, a significant result. There is indeed an enrichment of this blue, minor allele, little a genotype in the cases as compared to the controls. However, if we look at just the composition of the sample, we can see that that was just because population 2 happened to have more of this genotype compared to population 1, and this actually isn't directly related to the phenotype at all. So that's the concern for including mixed ancestry individuals in GWAS studies. So the way admixed groups are currently handled is generally by using principal components analysis. So here I'm showing a PC plot from the PGC PTSD group. You can see this kind of classic V shape for global diversity. So there's generally European ancestry on kind of one pole, African ancestry on another pole, excuse me, and then Native American, in this case, ancestry defining the third pole. So often the first step in large-scale collection efforts is to define who is homogeneous enough to include in a GWAS run. So we'll kind of draw a circle around where we have a dense amount of people who are homogeneous to each other and then exclude everybody else. And most of the time, this represents the European individuals in the collection, and so they are the ones studied. In the case of PGC PTSD, which is actually the most diverse of the PGC collections, we also had enough African ancestry individuals to do an African GWAS additionally. But this still means that all these folks on this cline are not sort of being utilized properly. So these are all these sort of African American individuals of varying fractions of European and African ancestry. So PCs can kind of tell you if somebody is, you know, 75% European, 25% African, or vice versa, but really just sort of this estimate of their, you know, global ancestry fraction is what's kind of going into control for their population structure. And there's actually a lot of fine scale structure that remains in the data. So I have this figure to try to illustrate this concept of local ancestry. So you can imagine if you, at some point in time, have two individuals that are each homogeneous for a different ancestry, in this case red and blue. Initially, their child will have one sort of copy of each you know, ancestral chromosome. And because of recombination, over the generations, these sort of long blocks of ancestry are going to get chopped up. So with more and more generations, more and more recombination events, these ancestral tracts will get mixed up to result in a kind of mosaic of ancestry that's going to be unique to each person, and the length of the tracts is going to be reflective of the demographic history of that individual. So what I think is important to note in this example is that both of these two individuals are 50-50 admixed, but you can see, you know, so on a global scale, on a PC plot, they would look the same. But if you zoom in on the local scale, you can see that they're actually clearly reflective of different demographic paths. They have different fine scale population structure. So the main motivation for this project was to develop a way to allow admixed people to be included in statistical genomic efforts in a well-calibrated way so that they could be analyzed readily alongside more homogeneous populations without any concerns about false positive results. So for this, I've developed a novel analytical framework, which we're distributing as a software package called Tractor, which accounts for the subtle differences in admixture actually at the genotype level. And this is how it's allowing admixed samples to be readily included alongside homogeneous ones. So I do this by incorporating local ancestry. So again, just to unpack that a bit, local ancestry tells us the origin, the ancestral origin of each tract in each admixed person. So in this example, you can see this person is three-way admixed. Their chromosomes are along the y-axis, and the position along each chromosome is on the x-axis. Because humans are diploid, the top and bottom halves are colored separately. And each person in the cohort will have a different kind of color mosaic depending on their particular ancestry contribution. So the intuition behind Tractor is that to correct for population structure in admixed people, we effectively scoop out the admixed tracts from each particular ancestry so that that kind of, you know, if there were a different minor allele frequency in these red European tracts compared to the blue African tracts, you know, by treating these individually, we'd no longer be confounded by mixing those two minor allele frequencies in an uninformed way. So we actually account for the ancestral backbone on which each minor allele is falling in our study. So circling back to GWAS, I was thinking of two primary use cases in designing this method. The first is the PGC-PTSD-like one, where we have one large-scale collection with predominantly homogeneous cohorts and then some admixed people that might be getting screened out. In that case, we could literally simply deconvolve those admixed people and analyze the respective ancestry components with their ancestral, cohorts. And we indeed in the Tractor software export basically just VCF files blanked out to only have the ancestral pieces from each ancestry with missing data for the rest. So this can be sort of readily done using standard GWAS procedures from there. We also had the use case of a large admixed cohort where we want to do a well-calibrated GWAS within it. And that required a bit of a more statistical solution. So for that, we developed a novel linear regression model that leverages the local ancestry context on which alleles fall to correct for fine scale population structure. So the statistical model, hopefully this looks familiar given the primer at the beginning of the talk. The statistical model built into Tractor tests each SNP for an association with the phenotype by splitting up the ancestry context of the minor alleles. So rather than just throwing into your GWAS, how many minor alleles are there overall, we're partitioning these onto which ancestry backbone they're falling on. How many minor alleles were falling on your African backbone, 0, 1, or 2, and how many were falling on European background, 0, 1, or 2. So that's, yeah.
How many ancestries can you do in one person? Can they have 3 ancestries that you look at, or 4?
That's a great question. And I think one of the nice features about this model is that it can extend to an arbitrary number of ancestries. So you can keep adding terms for however many more way admixed people are. So in terms of the statistical limitations, it can scale to an arbitrary number of ancestries. Practically, it can get a little tricky to call local ancestry on very, very multi-way admixed individuals. I've done thorough testing on 3-way admixed populations, and I have all the code and scripts ready to be implemented for that. Above 3-way admixed, I haven't done as much intensive testing, so I'd want to make sure that there is still good performance, but we can, the model can scale arbitrarily and the software can handle at least 3-way admixed.
Great.
Yeah. So yeah, basically what these terms actually are, to unpack this model a little bit. So again, we have our, we're predicting the phenotype at each SNP. We have our intercept term. This first term is the number of copies of our index ancestry at that position. So let's say the African is our index ancestry. This would say, do we have 0, 1, or 2 chromosomes that are blue at this spot in the genome for this person? This is kind of similar in flavor to admixture mapping. So you could get a effect size estimate for if it's just matters that you have an elevation of ancestry at this spot rather than the minor allele falling in that ancestry as well, which is a nice feature of this method as well. And then after that, we have these ancestry-specific terms that I was describing before, the number of copies of the minor allele on each component ancestry. You can also stick whatever other covariates you need after this. So you can put in all your clinical covariates, and we do also recommend putting in some estimate of global ancestry fraction as well, in case that's going to affect your phenotype. So I'll go over a lot of the benefits of this model throughout the talk, but one of the things I'd like to point out while it's on the screen is that we generate ancestry-specific effect sizes and p-values. So you can actually, you get a value for both, is this variant significant in the African genetic background, in the European genetic background, and they can have, different patterns, and often do have different patterns of association and effect sizes. So since we rely so heavily on local ancestry calls, I initially just started off doing simulations to make sure that local ancestry inference was accurate. Since we're running out of time, I might sort of gloss over some of the details of the simulation framework, but suffice it to say that I took homogeneous real data from European and West African individuals and admixed them according to a demographic model, which would give me a truth data set that looks like modern day African American, in this case, individuals. And then I could use that to quantify how well we're doing at painting our chromosomes and calling local ancestry, in addition to how well Tractor was performing from a power perspective. So maybe after the talk, I'd love to go into the details of the simulation framework, but I'd like to sort of gloss over that to get to a little bit more into the results. So one clear benefit of the Tractor model over traditional GWAS is that we see a power gain across many genetic scenarios in admixed cohorts. So effectively, if there's any difference across the ancestries, whether this is in the marginal or causal effect size, in LD, in NAF, we can see a boost in power using Tractor over the traditional GWAS model. So these different colored lines represent different genetic scenarios, but in all cases, the dashed line is our Tractor model, and the solid line is the standard GWAS model. So you can note the dashed line in these three settings is always above the solid line, and we can get pretty substantial power gains depending on the context. So in particular, I'll highlight this red situation where we have a minor allele frequency difference across ancestries. So just sort of to hit this home a little bit, if we follow our favorite, power level, let's say 80% across, using Tractor, we'd be able to find variants that have an odds ratio of, 0.1 lower. So there'd be the opportunity to find many more risk variants that would be impossible to discover using standard GWAS methods. And we did a bunch of tests to evaluate the landscape of when Tractor gives the biggest power gains, modifying the effect size, difference in effect size, absolute and variable math, admixture fractions, disease prevalence, sample size, lots of different features. I'll refer you to the manuscript to see the full spectrum of these. But I'd like to highlight two situations that had the biggest effect in Tractor power, which were a minor allele frequency difference across ancestries, which I showed one example of in the last slide, and here at different points in the allele frequency spectrum, I'm showing in panel B here. And secondly, when there's a difference in effect size across ancestries, and specifically this sort of interaction with admixture. So the most dramatic power gains that you can see, for example, in D, is when you have an effect that's only seen in the rarer ancestry fraction. So in this example, we are modeling a realistic African-American cohort. So that's, this model was about 80% West African ancestry, 20% European ancestry. Using vanilla GWAS, you would have actually no ability to find this risk variant. But using Tractor, we can get to, pretty respectable power thanks to basically deconvolving these ancestry pieces. So the kind of reason that we're recovering this power is that, if your signal is only present in about 20% of your sample, this other 80% would normally kind of swamp it. But by deconvolving the pieces, we're recovering the effective sample size of that number of tracks. And that's where this power gain is kind of coming from. I also think it's important to note that if there's no expectation for a benefit from including local ancestry, we don't see a dramatic drop in power. So in this situation, everything is identical across the ancestries. So, there's not expected to be any gain from having this extra level of information about local ancestry if everything's identical. We do see a small drop in power because we're basically losing a degree of freedom with this extra term in our model, but it's not so bad. So in no case did we see a dramatic drop in power of Tractor over traditional GWAS. Very quickly, I'll note that we did testing against several other models, including ASIMAP, which generates ancestry-specific effect sizes, but does not include local ancestry directly. And I also wanted to build in levels of local ancestry inference accuracy. So those figures I showed you were assuming perfect accuracy of calls every time, but that's not always the case. So I'd found in realistic situations for African Americans, 98% of the time calls were correct. So this is our kind of realistic Tractor performance in the dashed line, and then a lower bound of 90% in the dotted line. So the three black lines are Tractor at varying accuracies. Standard GWAS is in red, and then this other method in blue, and you can see again and the same 4 models that I presented the previous slide, we indeed still outperform them even at this lower accuracy level for local ancestry inference. We also quantified the type 1 error rate, the tractor model. Basically, we simulated a variant with no effect and saw how often it was genome-wide significant. And across our tests, we never saw this deviating from the expected false discovery rate of 5%. So we don't see any elevation in false positive rates with the tractor framework compared to expectations. So this means that this sort of observed power increase that we've seen is, due to finding true biological signal and not just false positives, which is always heartening. And we finally wanted to confirm that not only did we add power to GWAS, but that we were also doing a good job at estimating our ancestry-specific effect sizes, since this is a nice feature that we output from the model. So indeed, for every model tested, we produced highly accurate ancestry-specific effect sizes. So here I'm showing the simulated effect size for each ancestry as lines, and in all panels and throughout this talk, African is in blue, European is in red, and then the tractor estimates of the effect size at each point for these simulations are these box plots, and you'll see that they very, very closely follow the simulated effect sizes. So we're highly accurate in effect size estimates. So tractor seemed to be doing a really good job in simulation. So finally, I wanted to make sure that it also performed well on empirical data. So for this, I used about 4,300 two-way admixed between Europe and African individuals from the UK Biobank. And I ran some blood lipid biomarker phenotypes. In this talk, I'll only describe total cholesterol. The first finding, which is just sort of a confirmation, is that we were indeed able, with the tractor GWAS, to replicate all of the top hits from standard GWAS analysis. So, just comforting that we were indeed able to find what we should have found. Additionally, I'd like to point out that we also find some novel hits that would have been missed in our African tracks runs of tractor. So again, here's our standard GWAS on the top, tractor GWAS on the bottom, and I'll highlight this hit on chromosome 15, which was not seen in the standard GWAS analysis, but that looks very believable in our tractor analysis. There's also some functional support about this gene that would show a relationship to total cholesterol. So it indeed seems like a plausible new finding, thanks to studying the African component of these admixed people. Another nice feature of our model is that tractor is also able to better refine the location of GWAS signals to be closer to the causal variant than would be possible using standard GWAS procedures. So total cholesterol had been previously mapped to this gene DOC6 in some top med African American cohorts. So we wanted to investigate that signal here, given that DOC6 didn't really have a very clear association to total, mechanistically, there wasn't a clear path to affecting total cholesterol. So here's the results from our standard tractor GWAS, zooming in to this gene DOC6. Our top hit using standard GWAS for this phenotype is very close by to the previous top hit in the same intronic region. When we run our tractor analyses, however, our top hit is actually 20 kb downstream. And interestingly, this variant is, over DOC6 on the minus strand, but additionally is over this gene ANGPTL8, also known as lipase, on the positive strand. And there's some very clear functional evidence, supporting lipase as being very related to total cholesterol. It's been shown to regulate blood plasma lipid levels in mice. It's had its expression correlated with plasma lipids previously in humans. So basically it just suggests that this is a more promising candidate gene. Also interestingly, the variant identified by tractor is actually a missense mutation, and is the same missense mutation that was pinpointed in fine mapping studies in another diverse or non-European collection, Biobank Japan. So we actually found the first time around, just using our tractor analyses, the putative causal variant without doing subsequent fine mapping. So it's kind of exciting that, by leveraging these different LD structures in the different ancestral components of these admixed people, we can sort of the first time around get closer to our causal variant. So with that, I will wrap up. I just want to remind you of the primary deliverables of Tractor. So firstly, we've developed a readily implementable framework to allow you to include admixed people in large-scale collections. That was the primary goal. You don't have to toss admixed people out anymore. And beyond that, we do a much sort of finer scale correction for population structure. We also improve long-range phasing in admixed people. I didn't talk about that here, but you can read about it in the Tractor bioarchive. And then with respect to GWAS, our novel local ancestry-aware GWAS model can improve your results in a variety of ways, notably through boosting power under a variety of genetic scenarios. It generates ancestry-specific effect size estimates and p-values, so you can identify new ancestry-specific hits. It does a better job localizing signal. And as I said, these sort of ancestry-specific effect size estimates are very important output and can be used in downstream efforts like generating polygenic risk scores. So with that, I'd like to point everyone to the Tractor manuscript if they're interested to read more. Hopefully this will be published in the not too distant future, fingers crossed, but everything's already freely available online on my GitHub, and I would love to take some questions.
Thank you so much, Elizabeth. That was a wonderful description of the work, and just so exciting to see that it's out online now, because when I started at the Stanley Center 5 years ago, Julio told me that as an admixed individual, my results would not be possible to GWAS at that time. I was so devastated. And then—
well, no longer.
No longer. And I remember you telling me about this work at an early stage, and it really lifted my happiness level at the thought that at some point I could contribute to a study. So I think this work is going to bring a lot of hope and happiness to a lot of people who can now participate in research where previously they could not. So I see there's one question in the chat from Claudine, which is basically a variant to function type question, which you alluded to the importance of, analyzing GWAS data to understand what causal variants are. And then Claudine's question is around how do we get to directionality of effects? So whether you're seeing an upregulation or downregulation of the gene, if it is a gene, how can we use this information to help develop hypotheses about the role of particular genes in a disease trait? So
yeah, tricky and complex questions. Yeah, it's a very hard point to get from GWAS results to actual biological function. I guess maybe just broadly speaking, some of the post-GWAS analyses that are often run, maybe thinking more broadly about, rather than, so there's sort of, I guess, I visualize it as two ways, zooming really close into a hit with fine mapping and figuring out, okay, here's the potentially causal mutation, and then you can, in collaboration with bench scientists, dive more deeply into what that mutation does, using model systems or cell lines or other things outside of my realm of expertise. Or you can sort of think about hits more broadly in terms of looking at the whole collection of GWAS hits. Are there enrichments in particular sets of genes? So that's another strategy people will often use. You'll sort of see, okay, we tend to see more significant associations in genes of this biological, type of biological function or this biological ontogeny, rather than genes of this other one. And that can kind of help sort of collectively inform the kind of biological system that's being, probed with that analysis at a sort of a bigger picture scale. Hopefully that.
Yeah, and I'm remembering right now, actually, Claudine and anyone else interested to take a look at Henrik Henrika Hein's paper with Mark Daly and many others, trying to find a way to predict which GWAS hits could be loss of function, for example, and especially in ion channels, which is relevant to the Stanley Center. So a bunch of different techniques there. And Morgan had asked earlier on, why is a QQ plot called a QQ plot? And Rachel Iverson responded to say Q stands for quantile. Yep. And I just wanted to flag that, see if you had anything to add to.
No, correct. I should have said quantile plot. My apologies for for neglecting to actually not just use the acronym. But yeah, that's correct.
A lot of these things have acronyms that, even when I learn what the words mean, don't always help me understand anything more informative. So PCA is another one, that principal component analysis, where I'm like, principal component?
Yeah.
What's that? So on that note, we have a slightly different plan in mind for the discussion section of this primer than we have done previously, because Elizabeth covered two different topics in her talk. One was a more general introduction to GWAS for essentially for non-geneticists, and then the tractor discussion, which I think is of interest to everyone, but perhaps people with a genetics background might have deeper questions to ask you, or more nuanced sort of insights into the work that you presented. And so we're going to try something different this time, where in the main Zoom room, people who want to ask Elizabeth some more questions about tractor and admixed analyses can stay here. And then Hailiang Huang has very kindly offered to host a breakout room. And thank you, Hailiang. And I can say firsthand that he is an incredibly kind and gentle teacher for people without a complex genetics background. So for people on the call who don't know as much about GWAS and wanted to review some of the concepts Elizabeth covered, and maybe review some of them again, or ask some simple questions about, what does case control mean versus quantitative? I can't even remember now. Talk me through that again. Raise your hand in the participant tab, use the raise hand function, and we'll push you all across into a breakout room with Hailiang. And that provides you with a safe space to ask those questions. And we will see how that goes. And if it goes well, maybe we'll do more of this in the future. So we'll just give people a second to decide if they want to review the GWAS concepts with Hailiang, or stay in the main call to talk more about tractor with Elizabeth. And Samantha is our wizard behind the curtain, who's going to make the breakout happen.
Yes, so the first few people I've already moved over, and I will continue to move people as they raise their hands.
Awesome. So for those of you staying in the main call, go ahead and put on your videos. Everyone put on your videos if you'd like, just to join in. And for those of you who have questions, just feel free to unmute yourselves and ask questions without waiting to be called upon. I think we can just have a general discussion here. One question I have for you, Elizabeth, is what stage of the publication process are you at with the tractor manuscript?
Hopefully nearing the end. It is in second round review. At Nature Genetics.
Awesome.
So fingers crossed. It's been there for 2 weeks now. So hopefully nearing a decision.
That's awesome.
Hi, Elizabeth. This is Dome. I have a question about the new model. So when you split into different ancestry, is it necessary to include the principal components? Will that be overcorrecting for the population stratification?
Yeah, that's a great question. So we do think it's, we are recommending putting in some estimate of like global ancestry fraction, whether that be the first few principal components, or, if you actually ran like admixture, the program or structure, to get some kind of admixture fraction. It should be fine if we're just using the first few principal components, it shouldn't be a problem. If you're throwing in like, you know, a whole ton of principal components, we're starting to get a little bit worried about some issue of collinearity or something, but I haven't seen any issues. All of the tests actually that I showed today, all of the simulation results, were including 10 principal components, both for the tractor model and for the standard GWAS model. Sorry, I should have mentioned that in the talk. So yes, I think PCs are totally fine to include and are a good sort of way to get at that global ancestry fraction in there.
Thank you.
Yeah.
This is Bob Hansaker. I'm gonna come right behind that because this was actually my question as well. And can you talk a little bit about the intuition of why you also include global ancestry if you've already modeled local ancestry?
For sure. So yeah, I guess the main reason would be if there's some sort of relationship between how admixed you are and your phenotype. So if people at different points in that kind of cline of admixture are differing in their phenotype in a characteristic way, this could help account for that, in addition to, like, you know, whereas the local ancestry is really getting at that question of this minor allele on this backbone at this particular spot in the genome. But if there's sort of a systematic difference of just how admixed you are, that would also be affecting the phenotype. We want to make sure that we're correcting for that as well.
So would that be like an epistatic interaction that's ancestry specific, or is it like if your polygenic risk score overall was like highly correlated with your global ancestry or something like that?
Yeah, all of the above. Totally, all of the above. Could be some epistatic interactions depending, what amount of ancestry you have in the genome will affect those. Also, just in terms of, in terms of environmental factors too, like how admixed you are might affect your environment. Like, I don't know, all the examples I can think of are kind of dire, but like, I don't know, people may react to you differently depending on how they perceive you or something like that. And different people on the spectrum might experience things differently that way, or I don't know, not the best example, but just sort of to hit home the point that overall ancestry fraction could influence your phenotype in that kind of manner as well.
I see. But it's not trying to control for error in the local ancestry estimation.
No, yeah, no, that's totally different from that particular term.
Okay. Well, thanks.
Yeah, thank you.
So I have a basic question about how you figure out which ancestries people have. Do you, in the sample collections that you're working with, is there typically a question that people answer about what they declare as their ancestries that you use as an initial filter, or do you just toss that information out because it's so often wrong, perhaps? People, I usually, for example, I usually identify just as white on forms because I find the question really irritating and difficult to answer. But I'm half Latina with Native American ancestry also. And I'm like, I just don't want to get into this. So I'm just going to tick white,
I myself am also admixed, and I often just check other because I can't be bothered. Or
right?
Multiple boxes.
So do you even use that information, or do you just go straight for the genetics?
So in terms of the work that I have been involved with, we generally go straight for the genetics. Sometimes we'll do a cross check to see how people responded, but in terms of actually assigning people into ancestral groups, it's usually based on the genetic data. So yeah, often the sort of first pass will be that PC, principal component analysis plot. So you'll just sort of see how people fall on that plot. Maybe some studies will do an admixture analysis, so you can actually see the fractions in each person, and then designate people depending on how those fall out. In terms of the sort of second piece of that question, which was how we and correct me if I misunderstood, but how we actually assign the local ancestry. So yeah, that's how you could get kind of the global ancestry. In terms of how you get the local ancestry, for that, you need a bank of reference haplotypes. So we have a bank of haplotypes where we have sort of a priori assigned, here are our homogeneous European individuals, here are homogeneous East Asian individuals, here are like all of the various, and I've been at this talk, I should also have mentioned, while everyone was here, talking at the continental level, there's definitely subcontinental fine scale admixture that I'm really glossing over, but just speaking at the continental level, we'll have assigned our reference individuals, and then you run a random forest classifier in basically finding, assigning each of your tracks in your cohort data, matching them up with tracks in the reference population. So you can see, we know sort of in general, there's recombination breakpoints at these spots in the genome. So we're going to look in this area and like, does that match with more of the European individuals compared with a different reference category. So you basically just go through, tract by tract and paint each person's ancestry kind of that way.
Can I ask more questions about the Native American component? I don't know how much work you've done on that particularly. But I know there's going to be some Native American populations in the PUMAS grant that's ongoing, and I see Ben is on the call here as well. How similar or different are different Native American groups to one another? And I don't know how much information there is on each of these groups in terms of the reference populations that is that reliable information. Obviously with Europeans and even to a certain extent the African and African American groups, the reference genomes can be from many, many hundreds of individuals, but the Native Americans perhaps the much, much smaller sample sizes.
Totally, that is a great point and is something that, yes, myself and others have been running into a lot. There is really a lack of good reference whole genomes from, indigenous Native American ancestry people. So this makes it really tricky to call that ancestry component. So yeah, as you suggested, like we do rely on our reference panels. And so if there's something lacking in the reference panel, we're not gonna be able to assign it very accurately in our cohort data. So that is something of vital need. Most in terms of practically how I've been able to assign Native American ancestry thus far, given this sort of dearth of homogeneous reference samples, you can try to use admixed samples. So often like Hispanic Latino individuals are three-way admixed often, or at least two-way admixed with some Native American ancestry component. So you can sort of tweak your local ancestry inference model to try and identify admixture in the samples and use just the sort of Native American pieces. And this is an idea I've been sort of floating for a bit. I haven't done a super comprehensive dive into it thus far, but one idea I have been thinking about is trying to use these Native American pieces of Hispanic Latino individuals to try to cobble together a sort of composite reference genome panel from those tracts. It's a bit of a touchy issue, I think, given the sort of fraught history. So I haven't, I'm still kind of in the exploratory stages of this, making sure this is something that would be, both work and be amenable, I guess. Yeah, I think that's a really important question. Like if we don't have diversity in our reference panel, we're not gonna do as good a job here. And I noticed, Ben, did you wanna add to that?
No, you did a great job capturing that, Elizabeth. I think the only other things to add are the control of local ancestry information is really about, in some respects, getting at an estimate of what the allele frequency of a given variant should be. So it's sort of like, what are the chances of seeing this variant in this place? And there's a sort of complicated relationship between the global ancestry information, that there was also this question about inclusion in the context of those PCAs for control of confounding. And, you know, it's that control of confounding is good for soaking up phenotypic variants that may not, that may drive confounding at the specific place, even if the specific local ancestry information is doing a good job. But we're
of the axes.
The axis is in the chi-square unit, is related to the p-value. It's one-to-one mapping between the p-value. So for example, a chi-square of 10 is roughly
into the kind of control of potential bias in that space. I'm not, you know, I think there's probably another round of the methodological, you know, investigation under the assumption that you have comprehensive capture of variation.
Yeah, thanks for helping expand.
You have a couple more minutes left. Does any of the other participants on the call have a question for Elizabeth? Well, I heard a rumor, Elizabeth, that you might be moving to another position fairly soon. Is that right?
Potentially, I am on the faculty job market, so it's a weird year to be on the faculty job market. So there's, you know, some uncertainty, but I'll definitely keep you posted. And I do have some positive looking routes. So yeah.
Awesome. Well, good luck and keep us posted. And we're so incredibly appreciative that you took time today to give us the intro, which is perfect timing at the beginning of the year, and then also the details of Track2, which is super relevant. I should say that Ben will be hosting the next program meeting, and we'll be hearing more about the PUMAS grant with contributions from Latin America and Africa via NeuroGap and or other African populations also. Benjamin?
Potentially. A number of diverse populations from the Americas and Africa. NeuroGap is the vast majority of the African component, but there are some collaborative efforts starting up also in Nigeria.
Ah, that's right. No, that's awesome. Okay. Well, we're at 3:30. That was a terrific conversation, and more to come at future program meetings. Thank you everyone for your participation.